Hello everyone. Uh, Please look at this very carefully. This is about the nature of light, and also we call it as a absorption spectrum. Different plant pigments absorb the different wavelength of the light, and these wavelengths are not absorbed at the same rate. So, ability of the pigment to absorb the different wavelength of light can be measured with an instrument called as a spectrophotometer. So what is exactly the spectrophotometer contains? It contains the light, and we are already setting up in the form of the mission. Inside the mission, what happens exactly is light travels to the prism, which is called as a monochromatic light. Only one light we are passing. So because we know that VBGR colors will be present, first we will pass the violet color or indigo color, or any one light will be passing into that. So we call it as a monochromatic incident light, and the sample we are taking, it may be like chlorella or any kind of the things will be taking place. Then we will pass it on to this, and there is a transmitted light will be falling. Then photo detector will be present. It will detect amount of the carbon dioxide or amount of oxygen what has been released here. Amount of carbon dioxide utilized and amount of the oxygen released will be considered as a photosynthesis. Then it will be recorded on the screen, which is visible as a graph length, also nanometers. Then what is exactly absorption spectrum? A graph plotting light absorption by pigments versus wavelength is called as absorption spectra. If we look at this chlorophyll B, it is absorbing the violet color, indigo color and blue color. Then the least is considered as a red color. Then if you look at the chlorophyll A, it is absorbing the violet, indigo, blue color as well as the red color. Carotenoids are the observing only violet, indigo, blue, etc. So they are not observing much amount of this. The peak levels about this is very less in case of this. And action spectrum, the curve showing the rate of photosynthesis at the different wavelength of light is called as action spectrum, and it is prepared by illuminating the chloroplast with light of different colors and then plotting wavelength against the photosynthetic later, oxygen released or absorption taking place. Then when you are looking at this both action spectrum and also absorption spectrum, you are able to see this action peaks over here and absorption peaks here. Both absorption and action spectrum do not exhibit the complete one-to-one overlap, but match only at the peaks. It indicates that chlorophyll A is the chief photosynthetic pigment, and some photosynthesis also occurs at the other wavelength of the visible spectrum. And it is clearly shown that the more rate of the photosynthesis in occur near the blue as well as the red regions of the spectrum. These wavelength of spectrum are observed by the chlorophyll